Nerves tingle and tell us of what we are not. But what are we but nerves? All right, here's our buddy, Galen, the grouchy gladiator doc. And remember, at the beginning, I talked about how Aristotle said that nerves were controlled by and originated in the heart. And he was actually confusing ligaments and nerves. Whereas Galen, the astute anatomist who had access to some pretty butchered up bodies coming in from the Colosseum, disparaged Aristotle, disparaged those who know nothing of what is to be seen in dissection. I have shown in my book, available now on Amazon, on the teachings of Hippocrates and Plato, that the source of all the nerves, of all sensation and of voluntary motion is the brain, and that the source of the arteries and the innate heat is the heart. Yeah, there's more to the story. We'll explore more of Galen and Aristotle later. Galen didn't have everything right, of course, but the word itself, the word brain itself, is incredibly old. It goes way back before these people. It goes back before Latin, Greek. It is a Proto-Indo-European word, meaning it's a word that is so old it's before the radiation that had come out of Africa into the Caucasus, before some of them went east and some of them went west. It's an extremely ancient word derived from the word, I think you say that, mregmo. I don't know. I've never met anyone who can speak Proto-Indo-European, but I think it's pronounced mregmo, meaning brain or skull, which then became, in the Proto-Germanic language, ragnan, or brain. So the point of all that being that it was recognized early on that brains were extremely important organs, right? You can mess up some other organ in your body and the person can say, ow, that hurts. But if you smack someone in the brain with your club, then they're out. They're gone, right? So they recognized that the brain had a special status amongst all of the organs, all of the parts of the body. The word nerve, however, comes from the Greek neuron, meaning sinew or tendon, and thus reflects Aristotle's misunderstanding of the term, which is kind of interesting that the word comes from his mistake. So again, just looking at a couple of our other friends, Moses Maimonides, the court physician to Saladin, Moses Maimonides, the Israelite. And he also picked up on this theme that had been expounded by Galen, that one who does not know anatomy may mistake ligaments and tendons and cords for the nerves. Alessandro Benedetti, 300 years later, began to have a more careful understanding of the distribution of the nerves and didn't just see them as disconnected cords running throughout the body, but by means of the nerves, his description is by means of the nerves, the pathways of the senses are distributed like the roots and fibers of a tree. So he understood the branching pattern in them, and he also understood that it was sensation that was transmitted by them. Thus, if you cut a nerve, you lose sensation in the part of the body that had been supplied by that. Inherent to that, Benedetti realized that the brain itself was where the experience of that sensation rested, right? And then Da Vinci the quintessential Renaissance mind, said the medulla is the source of the nerves which give voluntary control, motion to the members. So this idea that the medulla oblongata, that first part of the brain, there's a tremendous number of nerves that are coming off of that part of the brain. And he's taking the first step into actual neuroanatomy and a functional description. So here's a beautiful picture where he actually delineates all this. And you can see in here, which is kind of interesting, we're not going to get a real chance to talk about it, but we can see here, these are part of the sympathetic chain ganglia. He drew the sympathetic chain along there. And so here he has the convergence of, oh no, these are the nerve roots that go into. Interesting. We won't really get a chance to talk about that, I don't think. This is what we will talk about. I'm going to talk about the hierarchy of the nervous system, just the CNS and the PNS. I'm going to define for you the difference between afferent and efferent, and we'll look at the different modalities. So nerves give us 4 kinds of information, and we'll look at what those are. And then we'll look at classifications of neuronal categories, so both functional and structural classifications. Then I'm going to talk about glial cells in the CNS and PNS. These are support cells, and we'll see why they're important. And then I'm going to begin to dissect the CNS just a little bit. We'll go over I'll remind you of the structures throughout them just a little bit, like we did in anatomy lab, and all leading up to this here, the discussion of sensory and motor pathways and how the motor homunculus works. That's one of the key concepts in the lecture where I want to get. And then I'll talk about the PNS a little bit. I'll talk about plexuses and dermatomes and what that means, and how that relates to the structure of the PNS. I actually cut most of this out just now while you were taking the quiz. I don't actually talk about any of that. And maybe a little bit of neuronal pools. I left reflexes in there because I think it's important. Most of this I cut out as well. So let's see what we get to. If we have time, I'll maybe talk about more. Who knows? So here's the basic structure of the nervous system. It's gathering information, it's processing information, and then it's acting on that information, right? So there's this sensory function where the PNS is pulling information up into the spinal cord and brain, and that's where the integration happens, and then the control comes back down through the same peripheral nerves. Here's a sort of diagrammatic hierarchy. So there's the central nervous system. It's made of the brain and the spinal cord. The peripheral nervous system really has 2 types of fibers. There's going to be somatic nervous system, which is primarily responsible for collecting sensory information and acting on the skeletal muscles. That's what that is, the somatic nervous system. Now I'm going to focus on just that. It's like, the central dogma of, the general somatic nerve. But the autonomic nervous system is also part of the peripheral nervous system. It's super fascinating, right? All the things that the brain controls in your body that you're not thinking about, right? You don't think to yourself, wow, it's a little chilly in here. Maybe I should go through some vasoconstriction in the peripheral arteries, maybe start doing a little bit of shaking, shivering to raise my body temperature. It's not something you're doing consciously. You're aware it's happening, but you're not pulling those levers, are you? And so all that stuff is the autonomic nervous system. And there's really 2 branches of that. the sympathetic nervous system, often sort of simplistically dubbed the fight or flight branch. And then the parasympathetic nervous system, again, simplistically dubbed the rest and digest part of the nervous system. I don't think I'm going to really talk much more about it than that. And then there is the enteric nervous system, which is like the jungle. It's like the jungle of the nervous system. Enteric, referring to your GI tract, right? So there's a huge number of neurons in the abdomen associated with the intestinal viscera. And it's actually one of the largest sources in the body of serotonin. Okay, so this is another way. So that's kind of a structural hierarchy of the nervous system in terms of like, okay, this part over here and that part over there. This is a kind of a functional category. And when you get to, certainly the ones in medical school, I don't know if they cover it in nursing school or not, but this is the way that the nervous system is categorized, all right? And I'm giving this to you. I'm not going to, and I'll use these terms sometimes, but I'm not going to like take this to the nth degree. We're not going to talk about all of these types of fibers. So the nervous system can be broken up into 3 dichotomies, all right? It can either be, there are parts of it that are general or parts of it that are special. And in general, that means that the general nervous system is like muscles and basic sensory information from your skin, right? Special things include like the senses, your eyes and your hearing and your smell, right? That's like the special part of the nervous system. And then there's somatic or visceral. And this dichotomy means somatic is basically like skeletal muscles, right? And visceral, like you can also think of somatic as being voluntary. It's the part of the nervous system that you are like super consciously aware of, all right? And then visceral is the autonomics. It's the viscera. It's the stuff that you don't have conscious control over, or not very good conscious control over. There are aspects of it. And then there's afferent and efferent. That's another dichotomy. So I think I talked about this in lab, didn't I? Did I talk about affect and effect? So the afferent, maybe I didn't. Here's some more language, English language for you. Afferent means something that's coming up into the brain, all right? To be affected by something, you are affected by something. Or you cause an effect. So efferent nerves are motor nerves. It's output, all right? It's fibers coming down from the central nervous system. And afferent are fibers going up. You get affected by something, or you cause an effect on something else, all right? That's what those prefixes mean. So all 3 of these combine, it's like 6 sides, 8 corners of the cube. They all combine for these 8 different fiber types. You can have general, somatic, afferents. So that would be like the touch receptors, the myocin corpuscles in my hand, for example. Or you can have special somatic afferent. That would be like my sense of sight, would be that. You can have general visceral afferent. This would be the feeling of hunger or something. That is like the feeling of something in your stomach that's coming. Special visceral afferent. I don't know an example of that. But so on and so forth. There's 8 different nerve fiber types when you think about functionally what they're doing. Okay? So along that hierarchy, we have afferent and efferent neurons, right? I talked about those nerve fibers. But there's also interneurons. Interneurons are in the central nervous system. right? Those other nerve fibers, all those 8 categories that I talked about, that's the peripheral nervous system. The interneurons are the ones in the brain, and actually, that's the bulk of them. That's the bulk of the neurons in your body are in the big squiggly worm that calls itself yourself. So the sensory neurons, there are 3 categories. There are interoceptors. These tend to be visceral neurons. They're going to monitor your GI tract, like your reproductive system, your urinary tract, things like that. And then various internal senses like taste, deep pressure, and pain, or nociception is what that's called. Exteroceptors, that's outside stuff like the surface of the skin, sense of temperature, sight, smelling, hearing. And then proprioception is the 3rd type of sensory receptor. Proprioceptors return information about joint position and the acceleration of joint change. So this is like when I'm doing this with my eyes closed, I still know that my body is in this weird position. That's coming from my proprioceptors. Or like LeBron James or something like that, who's like able to do amazing things in the air and slam dunk, right? That guy's got really good proprioceptive capacity. And then we said interneurons. So, this is like all the association neurons that come with like cognition and all the things that we think about a brain. And then there are the motor neurons. So there's the somatics that are the skeletal muscles and the visceral motor effectors. And these are the autonomics, the sympathetic and parasympathetic. And they have this structure here. There's a unique, there's 2 different strategies for the autonomics that has preganglionic and postganglionic fibers. I actually cut the slides out this morning that talk about that, but we'll get to it. Anyways, so when we look at the brain, again, these are morphologic ways of getting our hands, or sort of neuroanatomical ways of getting our sense of the brain and how it works. Again, this paradigm of like afferent nerves going to interneurons going to efferent nerves. We have these sensory receptors that are going to come into the central nervous system. If those sensory receptors, if those nerve fibers enter the brain directly, like come off the brain directly, we call them cranial nerves. All right? If they go into the spinal cord and then ascend, we call them spinal nerves. There are only 12 cranial nerves. And they do all kinds of things like your sense of hearing, your sense of sight, your sense of smell, etc., your sense of taste. Those are all things from the cranial nerves. But there's others, like the vagus nerve that goes down and like affects the heart, whatever. They do their stuff in the brain. They do their reflex center work in the brain. And then there's this motor output. And they synapse on muscles, on glandular tissue, on adipose tissue. Yes, adipose tissue is sensitive to output from the brain. Okay. So these sensory receptors in the brain are going to give us 4 types of information. Any sensory receptor, you think, well, it's just going to fire. It's going to, it's just sending the signal, like lights on, lights off. No, it's giving us 4 types of information. The first is modality. What is the mode of the information? What's the sensory mode? Right? Is it taste? Is it touch? Is it hearing, smell, sight? So that's mode. And I'm going to take as an example just the case of taste right here, the sense of taste. So fun. I did an experiment with the kids last night. I teach this class in the spring, biochemistry of food. It's just a fun class. But I was doing something like this with my kids yesterday. And I wanted them to know to see the difference between bitter and sour. We were talking about acids and bases, right? There are these 5 different sensations of taste that we have. 5 different sensations of taste. And it used to be, you look back in the 80s or 90s or something, and they would have these maps of the tongues, like, ooh, you just taste sweet right up here, but like salty's over there or something like that. That's bullshit. That's actually been debunked. And in fact, all of the places on the tongue that have these taste buds they have sensory neurons embedded in them that are able to sensate one of these 5 different types of sensations or characteristics about food, modalities. Okay? And the way the brain is able to untangle this is labeled line code, the labeled line code. So that just means that the brain has maybe this sensory neuron in a teeny tiny taste bud someplace here on the tongue. If you stimulate that one, the brain is like, ooh, that's bitter. Must be good for me. I don't want that. Or maybe it's salty, right? It'll stimulate this sensory neuron, but not any of the others. The brain interprets that signal as salty because this neuron, if you were to trace it back to the brain, it goes to the part of the brain that says, if this light comes on, this is salty. All right, that's all. It's just the labeled line code. The brain in each of these ends up in different parts of the brain. It gets sorted out. The point being that it gets sorted out at the brain, not at the tongue. It used to be that, if people thought this lights up, you only taste sweet here and salty there. That's not how it works. Labeled line code. Labeled line code is the concept that underlies all of sensation, but it's easily depicted here in taste. Okay. Second type of information is location. Where did the thing that you felt happen? All right. So this is, and this is anticipating where I want to go. Probably get there in like 25 minutes. Where I'm going to talk about somatotopic mapping and like how we have a sense of our body. There is a map in your brain of your body, and it actually kind of looks like your body. It's a map in your brain of your body. And again, the labeled line code works. When a neuron comes in to the brain, it goes to that part of the brain, and the brain says, oh, there's something on my elbow, or whatever. The thing is, and this is an important concept, because this, again, is going to play into the somatotopic map, is there are differences in the relative size of receptive fields. So our lips are very sensitive, aren't they? And our fingertips are incredibly sensitive if you don't have calluses. But our back, not so much, right? The skin of our back. That's because, I don't know if you've ever done this, but you can take 2 pencil points or something, and you can actually map out the size of a receptive field. So here's a very large receptive field that's being served by a single neuron. You can stimulate that skin anywhere inside that receptive field, and the brain's going to interpret it the same way. All right? So this person here, they only think they're getting poked by 1 point, not 2. All right? Because if this nerve triggers here versus here, it's the same thing. The nerve's just turning on. Large receptive field is inversely correlated with your ability to identify where something's happening. Whereas on your lips or your fingers, very small receptive field. high density of neurons. High density of neurons, small receptive field. So that same 2 points on your lips, you're like, oh, I got 2 points on my lips, right? Easy to tell. And this is because we use our lips. We use the skin on our face, right? You want to know exactly where something is, but particularly the lips or the hands, unsurprisingly, the external genitalia. But most of the rest of the body actually has a kind of a large receptive field, small density of neurons, low density of neurons. So low density of neurons in the back, high density on like the fingers and stuff. You guys got that idea? This is going to play in when we talk about somatotopic mapping. Third type of information is intensity. So, and when I say intensity, I don't mean how strong that action potential was, right? Because that action potential just is the strength that it is. If it fires, it's all or nothing, right? It just like happens or it doesn't happen. When I say intensity, there are these 3 ways that it can encode intensity. It can encode it by, this one's important, it's specifically which neuron fires, because not all synapses are the same, right? Certain neurons, the brain, in terms of, if you want to still think of it as labeled line code, it will interpret that neuron as being a really important neuron. If that one fires off, pay attention to it, okay? Versus another one like, For example, sensory receptors. Our clothes, you feel your clothes on your body, particularly autistic people, it drives them crazy because they can't ignore it. Most of us ignore it, but once you pay attention to it, you feel it, right? Our brain's like, it's not that important. We tune it out, right? But if you have like a pain neuron that's right next to one of these sensory neurons, that lights up, the brain interprets that as being important, right? You stick a needle like, oh, yep, sorry. How many fibers are doing so? That's another way that the brain can interpret the intensity of a stimulus. Are you like lighting up everything in this area or just one? And then the rapidity. So I told you that action potentials encode information in terms of frequency modulation, like how rapidly they're firing something. Okay. And then the last type is duration. And as the name suggests, like how long is that stimulus there, right? So this brings up the concept of adaptation and a couple other different types of neurons. There are slow adapting, or what we call tonic receptors, and then there are phasic or fast adapting receptors. So here, let's look at this picture here. We have receptor potential in this direction and time along here. So we're going to have a stimulus. Well, let's just start, I guess, with tonic one. You have a stimulus, and here's the stimulus. It comes on, we're stimulating it, and we turn it off. And that receptor potential goes up and stays up until the stimulus is released. Whereas a phasic, you have that nerve, stimulus comes on and stays on, but that membrane comes back to normal. It comes back to normal until the stimulus is released. And then we have a hyperpolarization, and it recovers from the hyperpolarization. All right? And so this makes sense if you think about certain things. So like, for example, I run my hands through my hair, get everything just right, and I felt my hands going through the hair. Okay, but like after I do it, I don't need to feel exactly how every hair is. Right, the hair didn't. I moved it, and I stimulated that nerve. Nerve has been moved, but it didn't like. I don't need to know that it's still moved, right? Just like, let me forget about it. And so that's a phasic thing. Whereas tonic is going to be giving me information about body position. So that like when I do this and my eyes are closed, I still know that my body is like this. I feel the stretch receptors in wherever. When I sit down, I still feel the chair on my butt, all that kind of stuff. Tonic receptors. So tonic receptors can be a problem. This can lead to, they can be the ones, so say you trigger a set of tonic pain receptors in your ankle because you've like sprained your ankle a bunch of stuff and it's just like sore, right? It's just sore and it won't go away. That's a tonic receptor kind of thing. All right. So here are some structural slides for Miles. Here are just looking at the neurons. Here are some different categories. This is the one we talked about today in lab, the multipolar neuron. Sort of easier to tell which pole is which from this cartoon. Not so easy in the actual slide. But the idea being that there are multiple poles and the dendrite signals come in and go through the cell body. There are anaxonic neurons. These are ones that don't really have an axon. They do have an output and an input, but you can't tell which it is just by looking at them structurally. So maybe these are all dendrites, and that's the output of the neuron. Who knows, right? And so the thing about an anaxonic neuron, I guess I could ask you, should I make you think about it? Where would you expect to see an anaxonic neuron? Like what kind of thing would it be doing? You don't have to get it, but it's fun to make you guys think. Well, the first thing, is it trying to send a signal anywhere? No, it's not. So an anaxonic neuron, it is likely to be one that's part of a complex cluster of cells that has a sophisticated processing job, where whatever that neuron's doing, it's like communicating to other neurons that are right next to it, right? And it's probably part of a complex tree in some nuclei in the brain or whatever. Then there are these bipolar neurons that are collecting information from one source, it's going through the cell body, and then it's coming out through the single source, as opposed to a unipolar neuron. You're like, wait a second, they look the same. No, they don't, because this is basically just a jumper cable. The signal that's coming in from the dendrite's not actually going through the cell body. Sadly, I talked about the perikaryon, and maybe I alluded towards the cytoskeleton and the effects that the cytoskeleton has in the brain, whatever. We won't talk about that in detail. That's a pretty high-level discussion. But just the effects of the signal coming through the dendrite and going through the cell body versus not going through the cell body has real functional significance in those cells. So those are the structural categories of neurons. Neurons firing, everybody? That's only half the story. Only half the story. The other half are glial cells. These are, just like here at Colby, you go home, you tell your parents ask you, how was school this semester, and you're like, oh yeah, it was great. I had these professors, this guy, Dr. Klepak, he was super fascinating, really excellent class. And then that's it, right? That's the end of the discussion. But frankly, you're all here. We're able to do this because Glenda cleans the building, right? She cleans the building. And like all the dudes over in the physical plant, they like keep the lights on. And like all those admin dudes that are, like scurrying around doing whatever they do over there. Everybody who does everything that they need to do, that's like the glial cells, right? They're not actually talking and delivering like academic stuff to you guys, but they're crucial for the function of the whole. So that's what the glial cells are. In the central nervous system, there's these 4 types. We have ependymal cells. Ependymal is how you say that word. And ependymal cells are, the job of ependymal cells is monitoring the cerebrospinal fluid. This is what the choroid plexus is made out of. The choroid is made from ependymal cells. So you're going to find this just on the surface of the ventricles. Then there are the astrocytes. So I showed you the slide of the astrocytes when I told you all that you had stars in your brain. And the job of the astrocyte is it maintains the blood-brain barrier. And it helps control the dissolved gases in the brain, and also gives the brain some of its 3-dimensional structure, and we'll see why that is the case when we look at the next slide. Then lastly, second to last here is the oligodendrocytes. Oligodendrocytes are like the Schwann cells of the central nervous system. They make the myelin. But instead of one cell doing one neuron, they do a single oligodendrocyte puts out projections and does several nerves adjacent. Microglia. Microglia are like the garbage trucks of the brain. They just monitor. They're phagocytic cells. Okay, so here is a little depiction. I told you in lab that when you see gray matter, you're thinking cell body, and when you think white matter, you're thinking axon or like a fiber tract, right? What do I want to show in this picture? So, yeah, I know. I'm sorry. This slide is like a combination of 2 slides that you guys have. It's like the same stuff. It's just like a little bit different. Why do you think in the peripheral nervous system you just have Schwann cells, which are the names of the glial cells that make the myelin in the peripheral nervous system out here in the nerves? And in the central nervous system, we have these oligodendrocytes that have multiple projections that can myelinate adjacent cells. I'll give you the answer, but I'm going to see. It's good to make you think deeply. Yeah, Miles.
Because the brain is like full of all interconnected, really complicated network. If you want an oligodendrocyte to maybe have like different parts of neural circuitry affect myelination of other parts of circuitry, I don't know any communication between 2 sites of oligodendrocytes.
Yeah, let's think of it more in just like a pure architectural perspective. You're on the right track. In the peripheral nervous system, they're just nerves. They're all traveling the same direction, aren't they? But in the brain, it is a really complicated, highly organized 3-dimensional lattice of neurons, right? Where nerves aren't all going in the same direction. But maybe you need to have a nerve that's going this way while this one's going that way. And so an oligodendrocyte helps create that spatial framework by connecting neurons that are not necessarily all just aligned and bundled up by the connective tissue or something like that. You see here the blood-brain barrier, the astrocytes helping to create that. All right. So I put this picture in. This isn't in your handout, but it's such a cool picture. This is a diffusion tensor imaging. This is a real image of a brain taken from a DTI. It's basically a a amped up MRI is what a DTI is. It's just it's an MRI technique where they're looking at axonal flow of fluid, right? So they're looking at the actual movement of fluid through an axon, through the axoplasm. And because of that, they can map out fiber tracks in the brain. It's like a way to look at white matter, and not just white matter as a whole, but like actually trace fiber tracks. So these are fiber tracks through the brain. You can see the corpus callosum right here, really high density. Here are the cerebral peduncles, that part of the midbrain. We talked about that earlier. And then all the projections out into the cortex. Pretty cool new technology. That didn't exist when I was your age. And then I put this slide in. This is also not your thing. I talked about this in lab. This is cool, though. I will upload this. I promise. This thing. I will upload it after. It's just showing that the white matter goes up with age, while gray matter actually goes down. And I already talked about the significance of that in lab. But just to show you that I wasn't making it up. All right. I find it to be interesting. Is there anything new here? Not really. This is just showing the process of myelination. I actually don't really care to go into that. All right. So then, in the peripheral nervous system, I've been mentioning Schwann cells. They are the oligodendrocytes of the nerves. They're these single cells that wrap layers of membrane around the axon. All right. So here is the axolemma that is being surrounded by the myelin sheath on the outside of it, and the very outer edge of that is what we call the neurolemma. So the only other glial cell in the periphery are satellite cells, and the job of the satellite cell is kind of like all-purpose maintenance. It's not a microglia. But it helps to monitor and maintain the proper dissolved gases and nutrient levels. It also facilitates neurotransmitter turnover. Okay, so we went through all of this in lab. The only thing I'll point out is just a few statistics. That's all. The brain is about a 1 to 2 liters, depending upon your age, and it's about 97% of the body's neural tissue is located in the brain. Interesting that it weighs about 3 pounds. Right? So the interesting thing about it, I have another slide that I blanked out here, but the interesting thing about it is that the brain is only about a few percent of the body's actual mass, but it consumes about 20% of your energy in the day. So the brain is actually the most inefficient of the part of the, I should say probably the costliest, energetically costly parts of the body. Brain has a very high metabolism. All right, so I showed you the ventricles, and now I'm going to talk about cerebrospinal fluid a little bit. We'll talk about circulation. So it gets produced by the choroid plexus, and it gets resorbed by something called the arachnoid granula. Now, the arachnoid granula, I think, I have a picture in here, is one of the 3 mothers of the brain. I talked about the dura mater, right? Well, there's the arachnoid mater as well, which is the spidery mother. Yeah, it's weird, but you'll see why they call it that, because it's got a spidery shape. What does the cerebrospinal fluid do? Well, it's it's like it floats the brain a little bit, right? So it's this fluid that it gives like some buoyancy to the brain because there's like displacement, right? So the brain is buoyant in the cerebrospinal fluid, and it acts as a shock absorber, a little bit of protection. Of course, it is sort of the broth of life because all the nutrients that the brain needs are going to be there. Lots of chemical messengers that are able to pass through the blood-brain barrier. All the dissolved gases and metabolic wastes, obviously, are also going to be in there. Okay, so here is the meninges. We'll talk about it again a little bit more. So here's the longitudinal fissure between the two hemispheres of the brain. The dura mater, the durable mother. Below the dura mater is the arachnoid mater. And we call it arachnoid mater because it has these like spidery little wispy projections that come down off the sheet of the arachnoid mater. The reason I talked about the superior sagittal sinus was in lab was so that I could show that here, this arachnoid mater goes up into, pierces through the dura mater into this sinus, this venous structure that's created by the dura mater. And this is where cerebrospinal fluid that was produced by the choroid gets sent up, gets returned to the venous blood system. All right. So, for example, people that are hydrocephalic, you've heard that term maybe before. It's children with really high intracranial pressure. That can either be because this gets blocked or clogged or is not functioning, or the choroid is producing too quickly. That's the other reason. And then the last layer, the third one, is the pia mater. Pia, little mother, the little mother. And that's this membrane that lays right on the surface of the brain, a thin little membrane right on the surface of the brain. So dura mater, arachnoid mater, pia mater. That's the meninges. So here you can kind of see a picture of the flow. We have some cerebrospinal fluid being produced here in the lateral and third ventricles, coming down through the cerebral aqueduct into the fourth ventricle, through the central canal, all around, surrounding the brain, braiding the brain, and then eventually being returned to the bloodstream via the arachnoid granula that are found along that superior sagittal sinus. Very cool. All right. So the blood-brain barrier, the 3B, it's made of the epithelium in the capillaries, right, of the actual blood vessels. The second layer is going to be astrocytes in combination with special ependymal cells that are the ependymal cells are there to help the blood-brain barrier in terms of producing the choroid plexus and the actual cerebrospinal fluid. All right. So this is where the cerebrospinal fluid meets the blood, and it's the ependymal cells that do it. These are all meant to be astrocytes that are helping to give the brain its three-dimensional structure. Okay, so that's the basic structure of it all. There are 12 cranial nerves. No, you don't have to memorize them. I'm just showing them to you. There are 12. The point here is that these cranial nerves have both sensory and motor fibers, both somatic and visceral fibers. All right. So Most of them are like special senses, so like sight, all the muscles that are focused on eye movement. There's a few skeletal muscles that get innervated. An important one is this one here, the vagus motor nerve. This will be important when we talk about the heart and how the heart functions. Vagus nerve meaning like vagrant. It's the wandering nerve. It wanders about the body and does all kinds of stuff. All right, that's all I'm going to talk about there. So moving down from the cranial nerves into the spinal nerves. This is the basic structure we talked about in lab. So we have a dorsal root and a ventral root, which come together and form a spinal nerve. And each vertebra is going to have, each vertebral segment is going to have a spinal nerve associated. Okay? So there's a C1 nerve root. There's a C1 vertebra. C2 nerve root, C2 vertebra, etc. And there are, throughout the different regions, as you're walking your way down the column, there are what are called plexuses. Plexuses. I'm not going to go through all the plexuses in detail. This isn't a full-blown anatomy class. But I'm going to point out, this one as an example of what a plexus is. A plexus is these nerve roots come off and then interconnect with adjacent nerve roots and form these highly branched, interconnected meshes of nerves that then finally irradiate into the terminal or peripheral nerves. All right? So it's just a way that nerve fibers from multiple vertebral segments or spinal nerve segments can integrate with one another. And you can imagine how fun it's going to be when you get to medical school and they actually make you memorize all that shit because they all have names. But I'm just showing you pictures of it. And the one I'm going to point out that's really important is the phrenic nerve, which you won't have to identify the phrenic nerve anywhere, but you should know that the phrenic nerve innervates the diaphragm. And what is the diaphragm? It's the muscle that helps us breathe, of course. And it comes off of C3, C4, and C5. So that means, what's the implication here to like traumatic injury? What do you think? Trauma. Somebody hurts themselves. Car accident. Like George Patton, the general from World War II, right? He got in a Jeep accident. He was sitting there, had broke his neck, hadn't completely, Yeah, if you sever the spinal cord at C3 or above, you cannot breathe, right? You cannot breathe. You lose the ability to operate the diaphragm. And that person needs to be intubated and put on artificial respiration immediately, right? So that's dangerous. Obviously, it make you quadriplegic, but quadriplegic people who can still breathe on their own, they have broken their neck below that level. So, all right. So what do you need to know in this picture? Okay, yeah. Just reminding you of the basic anatomy. There are the horns in the gray region and the columns in the white. So then, the way the spinal cord is organized, it's super organized. It's really, really highly organized. And in the gray matter, the dorsal or posterior portion of it, the posterior horn, is mostly sensory nuclei. All right, sensory nuclei are in the back, and motor nuclei are anterior. All right, in the anterior horn, and they're separated between somatic and visceral as well. So when these sensory neurons, these cell bodies, send out their axons that come out of the dorsal root, all of this is sensory. This is all sensory nerve here. This is all motor nerve here. They come together to form the spinal nerve, and they blend those fibers together. They mix the two. All right, and then they're going to branch out again after having come together. Yes, sir.
Posterior.
That's right. Yeah, right. So this would be the vertebral body right here, and then this would be like your inside your thorax or something. Okay. So that was the columns where the cell bodies live. Now we're going to talk about tracts a little bit. And there are ascending tracts and descending tracts. There are fibers that are going up, and there are fibers that are going down. So would you guess that ascending fiber tracts are afferent or efferent? Afferent. If it's going up, it's bringing information to the brain. Descending tracts are going to be efferent or motor. Just like some terminology here. Decussation, I already mentioned that word in lab. It's just the switching of sides from one side of the brain, the nervous system, to the other. And then the last, I think, of the directional terminology for the semester is contralateral and ipsilateral. These are simply relative terms. Things that are contralateral are on opposite sides of the midline to one another. Things that are ipsilateral relative to one another are on the same side of the midline. All right, contralateral and ipsilateral. So a neuron that's ipsilateral is not where the fiber tract that it's in is ipsilateral to the nucleus where the cell body is, does not decussate. And if the cell body is contralateral to the axon fiber tract that it's going to run in, it's going to have to decussate at some point. Does that make sense? That's the reason all those terms are there. You're looking at me, no. Yeah? Maybe? So if I showed you last thing the nuclei that were where the cell bodies live in the gray matter, and I'm showing you tracts now. If that cell body has a fiber that's going to run along some tract that is contralateral, it's going to have to decussate. That fiber is going to have to go across the midline, right? But if the fiber tract that cell body is going to run in is ipsilateral, then it doesn't decussate. So, okay. So this is. What do I have? A half hour. Okay. This is the beginning of my talk about the motor homunculus and somatotopic mapping. All right. We got to start by talking about the sensory pathways. So the sensory pathways that are going up to the brain, there are always three of them. There's always three of them. There's a first-order neuron. The first-order neuron is the one that actually gathers the data, gathers the information. It's the one that's on your skin somewhere, or it's on your tongue, or wherever it is. It's the one that's actually collecting the data. It's being stimulated. It's going to come in a normal somatic, so like a GSA fiber, a general somatic afferent fiber, like something that's telling me a bug's crawling on my skin or something. A GSA fiber, that cell body, the first order neuron cell body, lives in the dorsal root ganglion. That's where the cell body lives. it enters, and then it synapses on a second-order neuron. Second-order neurons live in the central nervous system. They live in the central nervous system. The first-order neurons are the only ones that are in the PNS. Second-order neurons live in the central nervous system. Their cell body is somewhere in one of these horns. Okay? It's in one of the nuclei in a gray horn someplace. That neuron, maybe it decussates, maybe it doesn't. I think usually they do. It decussates through one of the commissures and then ascends some fiber tract. Don't worry about the name of the spinal thalamic tract. Don't worry about it. But that's the name of it. Maybe worry. Well, don't worry about anything. But maybe think about it because it does tell you something about this next bit, and that is that the third-order neuron, the nucleus of the third-order neuron, lives in the thalamus. It lives in the thalamus. So that's where the synapse is. And then the fiber projects from the thalamus out into the cortex. All right. If this doesn't happen, if this synapse here doesn't happen, you don't know that it happened. All right. So for any sensory stimulus to be aware, for you to be aware of something, it has to have a synapse in the thalamus. Right? That's an important thing. So, well, how would that not happen? Right? Maybe there's some sort of reflex that's happening where this inner neuron has a collateral branch that's coming off, and the efferent. It's just a reflex. We'll talk about reflexes at the end of the lecture. Okay. Next, we're going to introduce Wilder Penfield. I think it's fun to, he's one of the guys I like. I like this guy. It's fun to talk about old scientists who were cool. This guy was really interesting. He discovered the whole concept of somatotopic mapping and what's called the homunculus. So he wrote this. He started at Princeton. He was on the rugby team apparently, and after Princeton, he went to Oxford and studied with William Osler and Harvey Cushing. I know those names probably don't mean anything to you, but there's a couple of you with doctor parents. You can ask them; maybe they know. But these two, the Osler and Cushing, were sort of like two of the very greats in terms of early 20th century and late 19th century medicine. They were like foundational fathers of it. We won't get into them. But he studied with them at Oxford and then went to work at McGill in the 50s, which is up in, Toronto? And while he was there, he was doing brain surgery. And he realized that he's like in there doing brain surgery. The brain itself doesn't have any pain receptors actually in it. So you can take the top off of somebody's skull and start poking them in the brain, and they don't say, ow. You can actually have them awake. Like local anesthetic around the incision. And he was doing these surgeries with people awake and stimulating parts of their brain, and they would say, I feel something in my finger. It feels like you're touching my finger. But he's just poking in their brain. Or he'd poke in somewhere else, and he's like, oh, I feel that on my cheek. Or I feel that on my shoulder. He mapped out the sensory map of our bodies on the brain by asking people while they were awake, and he was doing brain surgery on them by sticking little electrodes in their brain and stimulating parts of their brain. And he did this with sensory and motor. So he mapped all the muscles in the body as well. Anytime you're interested in reading about you're reading about his account of it. It's a super enjoyable book as well. I'm just throwing like fun medical reading at you. Yeah. The Mystery of the Mind. It's old, right? Like it was like from the 60s now or whatever, as you can tell by the cover, but it's pretty interesting. So what did he really discover? Let's look at the sensory homunculus. So it's this functional map of the sensory neurons in the body. And we find it in the post-central gyrus. So this thing right here has this map of your body on it. This map of your body laid out along the post-central gyrus. So when you look at it, it doesn't exactly look like a person, though. And the proportions are a little bit off. That's because the area that's dedicated in the post-central gyrus, or the primary sensory cortex, is directly proportional to the density of sensory receptors, right? So that should make sense to you, because if there's some neuron, it has to end up in for it to have a unique function, it's got to end up in a unique place. Our fingers, our lips, there's a lot of neurons here, right? There's a lot of neurons. So it's going to take a lot of area in the brain to map that part. So when you look at that somatotopic map, let's go back and look at this thing. This is what our brain thinks our body looks like, just spatially, if you wanted to lay it out, okay? We have a ton of area dedicated to our hands, a ton of it dedicated to the tongue and lips. That's the sensory homunculus, and very little dedicated to the back and legs and et cetera, the other things. Okay. This works with the motor, on the motor side as well. So this is the sensory neurons. These are the motor neurons, right? The motor, primary motor cortex, I guess is the right way to say it. And there's another map right there of the body in terms of the muscles, the motor units that are being controlled. So the area of the primary motor cortex is directly proportional to the density of motor units in that part of the body. We have a really high resolution of control on our fingers. So our motor units that control the muscles in our hands don't control huge chunks of muscle. They control very small, like a motor unit may only have a few dozen fibers, muscle fibers in it, in the hand. Whereas the muscles in the leg, one motor neuron may control a pretty big chunk of muscle fibers. So the density of motor units is low. High density motor unit in the hands, in the tongue and mouth, because we speak. This is where I should show. I was showing the TAs this video of people talking, and they had there's an X-ray video of a person talking, so you can see what the tongue and the inside of the mouth is doing when you're talking. It's pretty crazy how much control that we all take for granted in terms of the tongue and larynx and everything. So the point, anyways, being that the area of the primary motor cortex that is dedicated to a function is directly related to the density of motor units. All right. So this line, that line there, memorize that one, please. That concept. So now we're descending. We sensed something, sent it up through the 3 neurons to get upstairs, processed it through the map, sent it over to the muscle map, and now we're going to send it down. And it comes down through one of 2 pathways. It's either, we'll get to the 2 pathways. Whatever pathway always involves 2 neurons. 3 neurons to get upstairs, 2 neurons to get down. One of them is, the first of them is called the upper motor neuron. This is a GSE fiber that originates somewhere in the primary motor cortex and is going to come down and synapse somewhere in the central nervous system on another motor neuron, the lower motor neuron, depicted in black here. The lower motor neuron is the one that's going to emerge from the central nervous system. If the lower motor neuron is coming out through a cranial nerve, all right, if it's coming out through a cranial nerve, it is not decussating. It's not decussating. If the lower motor neuron is coming out through a spinal nerve, that upper motor neuron decussates. All right? Who cares about that, right? I mean, like, get out there and do your job. Like this, left side, right side. No, there's real significance in this because, and let me tell you this, there's a little clinical pearl embedded in this super esoteric detail. And that is when a person has a stroke, when a person has some kind of lesion in the brain of some sort, you can identify with remarkable accuracy without having to do a brain scan. You can identify with remarkable accuracy where that lesion is dependent upon the pattern of pathology that you're seeing in the person's body, right? So there's upper motor neuron presentation versus lower motor neuron presentation. Say you have a lesion up here in the primary motor cortex, right? You're going to have face drop on one side and weakness in your arm on the other side. Okay, you see that? Whereas, say you have some kind of lesion here, it's going to be a lower, say you have some sort of lower motor neuron syndrome. Lower motor neuron syndrome would have, you could have the pathology localized to one side. So the point being, and I don't want to get into this like super fine resolution details of that point because I'm not testing you over this. The idea is simply that the upper motor neuron decussates or does not, depending upon exactly where it's emerging from the central nervous system. Okay? So maybe you'll think of that in the future sometime when you walk your way further down the clinical path. I already talked about this. I talked about this already in lab. This is just the layers of the connective tissue that make up a peripheral nerve. I'm gonna keep trucking. Okay. There are 5 nerve plexuses. These are they. I'm not gonna ask you to memorize all those. I did talk about the cervical plexus. But the reason I'm showing these is to show you that the body the peripheral nerves are mapped as a function of the nerve roots into what are called dermatomes. So if you look at the sensory nerves that supply your body, there's this sort of zebra pattern that goes across the body that relates to the different spinal nerves. And you can see the posterior and anterior of all that. This is important for a number of reasons. Again, so, you may be thinking in this age of like high resolution imaging, why does a clinician need to be able to like identify something like exactly in the office? Why can't they just send them in and get radiology and see what the radiology? It's important to be able to direct it's important to be able to direct the care of the person as quickly as you can. So if you get a sense of what's happening, the sooner you can get a sense of what's happening to a person, the better the outcome is going to be for that patient, right? So understanding the anatomy and is going to do nothing but help your clinical skill. All right, so what's one of the clinical significances of this? This is an example of shingles. So all of you probably had the chickenpox vaccine, I hope. Yeah, I didn't when I was a kid because I was a kid in the 70s. And I got chickenpox, which is not super fun, but it's the herpes zoster. It's in the family of cytomegalovirus. Most people have come in contact with this virus. What happens is if you got chickenpox as a child, the varicella, which is the actual virus, varicella, it's in the family of herpes virus. It's not exactly the same virus that causes cold sores, etc., but it's the family. It lies dormant. It lies dormant, specifically is able to evade the immune system because it lives in neurons, in the cell body of the neuron. And the thing about neurons is they have what's called immune privilege. So your immune system is constantly monitoring all the cells in your body to see if there's something wrong. Is that cell infected by something? If it's infected by it, if it's not that important, let's just kill the cell, get rid of whatever was inside of it. Neurons are immune privileged because the body doesn't really want the immune system going on just like killing off all your neurons just because they've been infected by like the varicella. So varicella is able to evade the host immune system by localizing in this immune privileged space inside the neurons, sensory neurons. The problem is every so often, particularly when you become stressed, when cortisol levels go up, you can have an outbreak of varicella where it's not chickenpox any longer, but you have these eruptions that present themselves on the skin, all right? So, and that's what we call shingles. You can get the shingles vaccine that can prevent this, or at least lessen it. But what's interesting about shingles is that it shows itself along the pattern of a dermatome. So it's living in, when you have an eruption or a breakout, it's going to be from a single nerve root, right? In the dorsal root ganglion, it's those cells in the dorsal root ganglion that get infected. And you'll see this pattern of a rash. If somebody comes into the clinic, they have a painful, it's painful, it's not itchy, it's a painful rash that is only on one side, and it matches this dermatomal pattern that you see right here. That's shingles. Okay. Oftentimes, before you even get the eruptions, there'll just be pain, like real pain along a dermatomal pattern. Questions? No. Pain in general is a really interesting topic. Here are just some patterns of referred pain. So this is visceral pain now, not so this would be GVA fibers, general visceral afferent fibers, that would be reporting on like whatever your liver or your appendix or all these different organs that are in here, the heart, lungs, stomachs, pancreas, whatever. The thing is, because we don't really have a map in our brain for where our liver is, because we have no experience of where our liver is, those nerve fibers, they sort of piggyback onto the actual map of our surface body that we have. And this is what we call referred pain. Referred pain. So the liver and the gallbladder, someone has cystitis, which is an inflammation of their gallbladder. You feel it up here in your neck. What the hell? It's just how it got wired up. It's how it got wired up. So you feel heart. I mean, we've all heard about heart pain that happens over here on the left side. That's referred pain. What are some other examples? The kidneys. So kidneys can cause pain all over the place. All over the place. This whole lumbar and the loin and the gluteal region over here along the IT band. Yeah. What other interesting ones are there? Often they make sense, though. You know, like the pancreas localizes to the skin right above the pancreas. Referred pain. Strong visceral pain can stimulate neurons that are part of this spinal thalamic path. So that's as far as I actually was hoping to get, and then the rest of this is just gravy now. I'll start into the gravy zone. See how far we get. Are there any questions about all of that? That's like the super basic functioning of their nervous system. No? Okay. So let's get a little bit more interesting, maybe. There are two classes of neurotransmitters, excitatory neurotransmitters and inhibitory neurotransmitters. Excitatory neurotransmitters are going to be ones that open up sodium channels, right? They're going to open up sodium channels and drive that neuron to threshold. Inhibitory neurotransmitters are going to be ones that cause a hyperpolarization and further make it drive the membrane potential away from zero. Okay, and this can be this is typically chloride channels. So you had asked about chloride. Who asked about chloride? It was you, one over here. Yeah. These inhibitory neurotransmitters, there's different types, but often they're chloride channels. We'll see some pictures of that in a sec. So here we go. This is just a general overview of the categories of neurotransmitters. Chemically, they take a lot of different forms. Here's acetylcholine. So on the end, here's that acetyl group, just like acetic acid, right? As if the CH3CO2, and if the rest of this was an H, that'd be acetic acid. But here's an acetate ion, but it's attached, it's an acetyl group attached to choline. That thing is choline. The acetylcholine esterase, this is an ester bond right here. When you take an acid and you take the hydrogen off and attach it to a carbon, that makes this an ester. So the esterase cleaves that ester bond when acetylcholine is being turned over. That's all by the by, right? I'm not trying to get you to memorize it. But that's acetylcholine. There are also various neurotransmitters that are based off of amino acids. So GABA is an amino acid. Glycine, so GABA is a chloride channel neurotransmitter. It's inhibitory. Glycine does have some use as a neurotransmitter, aspartate, and then glutamic acid or glutamate. Glutamate is, in the beginning of the lecture, I talked about the taste modalities, the 5 tastes: sweet, sour, bitter, salty, and umami, which is is savory. Umami, that taste, is triggered by glutamate. It's a glutamate receptor, right? The umami sensation that we have, that like super savory taste that is in a lot of, well, any kind of food that you like, but a lot of Asian food knows how to use glutamate well. That's a neurotransmitter that's derived from. And then there are these various other monoamines, all of which, is that right? Yeah, they're all derived from amino acids as well. but usually tryptophan. Serotonin and histamine and melatonin, those are all these are just various biogenic monoamines. Dopamine, norepinephrine, epinephrine. All of these structures are chemically related. And then lastly, there are these neuropeptides that have some use, like your endorphins. Everybody likes the endorphins. That's a neuropeptide. Different categories. What do I want to say? I think that's it. So then, here are the different types of fibers and receptors and fiber pathways. So this is a little bit of autonomics. I had this slide was on the website, right? I'm sure it was. Yeah. I'm skipping over a lot though. You'll notice I cut a lot out. Parasympathetic nervous system is, so if you have a rest or digest, right? Like if your parasympathetic nervous system is really going, then you're said to be having sort of a cholinergic wash. These fibers, these are all cholinergic receptors. But the one that we have in parasympathetic is called a nicotinic receptor. So these are receptors that are triggered by acetylcholine, also triggered by nicotine, which is, I guess, one of the reasons that smokers find smoking to be calming. I don't know. I kind of pulled that one on my butt there. These are excitatory receptors. Then in the sympathetic nervous system, there's a couple different strategies. There's either adrenergic or cholinergic fibers. Most of them are adrenergic, where you're having the preganglionic fiber is acetylcholine. The postganglionic one is using norepinephrine. Norepinephrine is a hormone that, amongst other places, is produced in the suprarenal gland. On the other hand, we have these cholinergic fibers that are going to use acetylcholine at both the preganglionic and postganglionic synapses. So in general, these are all efferent fibers. So you're having a preganglionic and a postganglionic, two fibers. There's always a synapse. In all of the autonomics, there's going to be a synapse that happens outside of the central nervous system. In the sympathetic nervous system, it happens in these chain ganglia, and the parasympathetic nervous system, it happens what's called paramurally, right next to the organ that fiber is innervating. I don't know. I feel like I'm getting lost in the weeds. Here we go. This is the inhibitory synapse that I was talking about. So GABA, the gamma aminobutyric acid, that comes in, will open this channel and allow some chloride to come in. When chloride comes into the cell, this is going to push that transmembrane potential towards a negative potential. And it makes that neuron less likely to fire. It hyperpolarizes that neuron. All right, I've got 5 minutes here. Let's see what I can actually talk about in 5 minutes. I can get through a reflex. Let's skip that one. Let's just talk about a normal, regular reflex. Step on a tack. Ow! And that's going to generate some stimulus. that's going to result in some sort of action, right? Well, we're going to lift our hand off of the pain. We'll see how that unfolds here. What does a reflex have to have for it to exist? So it has to have stimulation, right? You have to elicit, you have to have some kind of stimulus that's going to meet the criteria for eliciting that reflex. A reflex requires a fast, totally involuntary and stereotyped response, meaning it has to be stereotyped because you're not thinking about it. You're not controlling it. There's just like an automatic, quick response. Sometimes some things are so emergent that you cannot spare any moment while thinking about something or trying to have like higher order processes. It's just like right on the ground. You step on a tack. As soon as the body has evolved to this point where as soon as you get that kind of stimulation, we need to just pick the foot up and not keep going. Because every millisecond that you delay, maybe you'll step further and further on the tack, and nobody wants that, right? So let's see how this goes. We have the first order neuron. It's a sensory neuron pathway. It comes in and it's going to synapse on the second order neuron. Now before, I said that the second order neuron decussates and then goes up the pathway. But because of the identity of that first order neuron, it's a pain neuron that's telling me, wow, I'm stepping on something. It's going to have a collateral branch here that's going to synapse on a muscle, on a motor neuron, directly on a motor neuron. And it's going to turn that motor neuron on. Maybe that motor neuron has got some other fibers coming down from the brainstem, of course, or from the primary motor cortex. Of course it does. But that's not what's important. It's cutting all of that out and directly synapsing on that motor neuron. And this is the extent of the information processing. It's just super short little 3 neuron jumping through this reflex arc, and then it's going to activate the muscles. And that leads to you lifting your arm. Okay? So this is a neural reflex. There's all kinds of different ones. We're not going to go through all the steps in this, but there's the patellar reflex. Sometimes these muscle reflex patterns are triggered by stretch receptors. It doesn't have to be pain. There are these things called muscle spindle apparatuses that are in your tendons that if they get stretched, if they get stretched rapidly, it's about the acceleration of that stretch, then it'll cause some kind of muscle reflex arc to happen. So without going through all the details of that. They get quite complicated. Again, I don't want to go through the details of this, but so say you step on something and there can be an activation of the muscles that cause you to lift one leg, and also an inhibition of the muscles that would push that foot down. And it can also cross and then strengthen the muscles that would push that leg down and inhibit the ones that would pull that leg up. All right? So it basically you can have these very complicated patterns of firing that will elicit a coordinated response that will give you the kind of reflex reaction that is going to help with the situation. We don't need to go into that one. The point with this slide is the last slide. I'm right at time. There are not just skeletal, they're not just somatic reflexes, there are also visceral reflexes. So for example, you can have the carotid bulb right here, monitors. They're baroreceptors, pressure receptors. We talked about them when I was talking about homeostasis or something the first day of class. When I talk about somebody having orthostatic blood pressure and they stand up and the blood pressure drops and you go gray and all that. It's the same receptors, these baroreceptors here. And there's a reflex that goes on this reflex arc that comes down through the vagus nerve and can affect the sinoatrial node in the heart and change the heart rate. So, okay, that's everything. Epic day. There's the recap.